hello to all the enthusiastic students out there. This is your Prasanna Ma'am from NEET World. So, in this session, we are going to discuss on APEAPCET question paper Botany, which has happened most recently. So, to which set of students this session is really beneficial? This session is highly useful to all the students who have just landed onto your 12th standard, and this is highly useful to all the students who are going for a repeater course. So, from this paper we are going to discuss the concepts come the objectives, we are going to discuss the mnemonics which are used here and there, and we are also going to look into the memory hacks. Most importantly, we are going to look into the weightage of the chapter as well. So, stay tuned for other half an hour to 40 minutes. You will get exposure on so many topics and chapters here with respect to objective application also. So, yes, now let us get started with the paper of May 19th shift 1 2025 of APEAPCET, which is a very standard paper ma. Each and every student who are going to write APEAPCET or TGAPCET or NEET exam have to go through for sure. So, if you look into the first question, it is based on the herbarium, which is from the living world or diversity of from the living world, wherein herbarium sheets label provides the information about. Okay, this is from taxonomic aids of living world. So, if we have to look into what information do we get from the herbarium sheet, we will get the information about the date and place of collection, common name and family of the plant, and collector's name also. So, here we have to go with the option 3. Fine. Next, that is most generalized question, and this is from the taxonomic aids of diversity in living world. Second, so here this is important ma, look into this question more detail. This question is from biological classification. Okay, this is from biological classification and it is from the kingdom fungi, which is very important. Okay, and to remember all the examples from all the classes of fungi, all the divisions of fungi, have made a short also, you can go through that. Now also I will tell you, listen, among the following fungi which are placed under Deuteromycetes of fungi, he has asked. So, how many divisions are there under fungi? We have 4, right? We have Phycomycetes, and Ascomycetes, Basidiomycetes and Deuteromycetes. For Phycomycetes, to remember, the best mnemonic I gave is RAM, where R stands for Rhizopus, Albigo and Mucor. Where Albigo comes under Oomycetes, Rhizopus, Mucor comes under Zygomycetes. For Ascomycetes, best mnemonic is ACP and team. Shortcut, where A is for Aspergillus, Claviceps, Penicillium, N for Neurospora, Morels and Truffles, and unicellular fungi, that is yeast, Y for yeast. Basidiomycetes, best mnemonic is A pulp. Okay, Agaricus, Puccinia, Eustilago, Lycoperdon and Polyporus. For Deuteromycetes, it is damn easy, that is cat. Okay, Coletotrichum, Alternaria and Trichoderma. This is how you can easily remember all the examples which are given in our academy in just 5 seconds. Okay, so with this easy trick, now you solve this question which are placed under Deuteromycetes. It is as simple as it is. Okay, so it is Coletotrichum, Alternaria and Trichoderma. Alternaria and Trichoderma. So, these 3 are placed under fungi imperfecti, that is Deuteromycetes. So, here we have to go with option 1. Clear, right beta? Next. This question, if you have to look into, he is asking choose the incorrect statement. Whenever statement based question is given, you have to focus on whether he is asking you a correct statement or an incorrect statement. As such, now he has asked you the incorrect statement among the following. Okay, this is from basic unit where the statements are given as such, okay, wherein plant kingdom is also involved, plant kingdom chapter is also involved, statements picked from different chapters are asked here. The present era is called as golden era of biology, yes, true. Sexual system of classification is proposed by Theophrastus, wrong. We all know, right? Sexual system of classification is also called as artificial system of classification. Okay, this is also called as artificial system of classification. This is given by Linnaeus. Okay, Carolus Linnaeus, where specially, why is it called as sexual system of classification? Because Linnaeus has focused on the andrecium structures, right? So, this is also called as artificial system of classification, and the proponents of this system are Linnaeus and Aristotle. Fine, it is not Theophrastus. Next, study of amphibians of plant kingdom are called bryology. Amphibians of plant kingdom are called as what? Bryophytes, right? Bryophytes, synonym of it is amphibians of plant kingdom. They are called as amphibians of plant kingdom. This is also true only. Recycling of nutrients takes place by parasitic petroplants. There is no such term only, but recycling of nutrients, it is done by fungi because it has saprophytic mode of nutrition where it feeds on dead and decaying matter. So, here what are the two incorrect statements beta? 2 and 4. Okay. So, we have to go with the option 2 and 4. Fine. Next question if we have to see. So, this is again assertion reason question. It is very important to tackle assertion reason question. Look into it properly, where in assertion it is given like algae will increase the level of dissolved oxygen in the immediate environment. True, algae increases the amount of dissolved oxygen. Second, they form food for aquatic animals. True, algae is forming food for aquatic animals. Okay, because sometimes they are acting as phytoplankton also. Here assertion and reason both are true, but in reason it is nowhere explaining on how the level of dissolved oxygen is increased by algae. Okay, it is explaining one more statement which is irrelevant of assertion. That is why we have to go with the option assertion and reason are true, but reason is not correct explanation of assertion. Okay, reason is not correct explanation of assertion. Next, here you see ma, choose the correct statement. This is from morphology of flowering plants. Definitely we will get a question from the examples only from morphology of flowering plant, be it in NEET or APEAPCET TG or AP. You see, choose the plant from the given list which have hypogynous flowers. Okay, mustard, cucumber, brinjal, plum, rose, peach, guava, sunflower, China rose, this and all are given. So, here we have three types of flowers like hypogynous, perigynous and epigynous flower. Okay, hypogynous, perigynous and epigynous. Hypogynous with superior ovary, what are the best examples for hypogynous flower? Mustard, China rose and brinjal. For perigynous flower it is plum, rose and peach. For epigynous flower which has inferior ovary, examples for it are guava, cucumber and sunflower. Fine. So, here we have to go with, for hypogynous flower we have to go with the option mustard, China rose and brinjal. Fine. Next question ma. So, this is a very nice question which is application based. Look into it, you will really enjoy solving this question actually. Three plants he has given. Mustard, calotropis and guava are collected. There are 30 notes for mustard. For mustard, how many notes he is saying? Mustard has number of notes included are 30. Next, 25 notes in guava. Okay, how many notes in guava? 25 notes. And find the number of leaves, 25 notes, 30 notes in calotropis, 25 notes in guava. In calotropis, how many notes? 30 notes. Find the number of leaves put together in all three plants. So, number of leaves he is asking. Arrangement of leaves is described under the concept phyllotaxy, right? So, mustard comes under what phyllotaxy, Nana? Alternate phyllotaxy. whereas guava and calotropis put together, they are placed under opposite phyllotaxy. Okay, mustard under alternate phyllotaxy, guava and calotropis under opposite phyllotaxy. Alternate phyllotaxy, for at each node there is only one leaf found, right? At each node there is only one leaf under alternate phyllotaxy. So 30, right? For mustard, how many nodes are there? 30. So 30 nodes means 30 leaves. Here I will give you how many leaves. For 30 nodes it is 30 leaves. Next, guava and calotropis 25 nodes. At each node 2 leaves are found. Okay, at each node 2 leaves are found. If that is the case, 25 into 2 it is 50 and 30 into 2 that is 60. Okay, 30 into 2 60. So 60 leaves for calotropis and 50 for guava. On total, how many leaves he is asking? Find the number of leaves put together for all 3 plants he is asking. So all the 3 plants, how many beta? 0, 6, 140 total, right? Now if you clearly look into the options, see 140 is there right over here, that is the correct option. Next. Next question, if you have to see, this is match the following type of question, where sexual reproduction of flowering plants and cell division, both chapters put together, they framed one question. Look into it more clearly. First is vivipary. Vivipary means what? The development without detaching from the parent plant. Seeded plant which is developing on the parent plant without detaching, that is vivipary, right? So here vivipary is reducing the environmental stress for the seed to develop outside, okay, that is developing inside the parent plant only. So it is reducing the environmental stress and establishing a plantlet attaching to the parent plant itself. Second, parthenogenesis. Parthenogenesis is what? Development of embryo from an unfertilized female gamete. Okay, this is development of embryo from an unfertilized female gamete. Next is what? Meiocytes. Meiocytes are what, Nana? Meiocyte are nothing but the cells which are going to undergo meiosis. Those cells we will call it as meiocytes. So meiocytes in diploid organism, they are specialized to undergo what? Meiosis. Next is perennials. Perennials, best example is bamboo only. Bamboo flowering at the end of the life. This is the best example for the plant which is a perennial plant. So this is how we have to look into, okay, which is a combination of 2 chapters. Now if we have to look into the options, A will be 1, B will be 4, C will be 2 and D is 3. Fine. Next. Next question, ma. Here you see, match the following type of question. Pollination, this is also from sexual reproduction of flowering plants. Xenogamy, the pollination between 2 different plants is xenogamy. Okay. So here genetic variation will be there, because xenogamy, genetically different types of pollen grains will be received by the plant, because it is for 2 different plants. Ophiophily is pollinated by what? Snakes, that is ophiophily. Chasmogamous and clistogamous flowers, which are seen in viola, common pansy, okay. So it is seen in those flowers. Chasmogamous, opened flowers. Clistogamous, which purely conducts bud pollination, which is closed all the time, right. So here chasmogamous flowers, if you see, those are which have exposed anthers and stigma. Clistogamous flowers, where flowers will not open at all. Okay, we have a diagram in at the area where it is explained about geitonogamy, xenogamy, autogamy. There we have a diagram which explains you about clistogamous flowers and chasmogamous flowers in viola or common pansy plant, wherein chasmogamous flowers are placed above, wherein which has exposed anthers and stigma. Clistogamous flowers which are placed below, they will not open at all and purely they will conduct bud pollination only. Only autogamy is possible for clistogamous flowers. Flowers do not open at all. Very simple question. Next. This if you have to observe beta, directly it is asked, okay, from morphology of flowering plants again. The characteristics are given to us, we have to write down the floral formula. Very simple. Ebracteolate, how we will write? EBRL. Next, united perianth, perianth should be bracketed. Next, pentacarpellary, gynoecium should be 5 and syncarpus, so bracketed. Next, zygomorphic with superior ovary, so hyphen or stripe should be below the gynoecium. Bisexual, we have to write, we have to keep this symbol. Both male and female gametophytes are present. Fine. So, this is the floral formula based on this description. Next. Here if you see, this is from cell biology beta. Lysosomes were found to be very rich in hydrolytic enzymes. These enzymes are optimally active at basic pH, which is completely wrong. Lysosomes are found in, they have very rich in hydrolytic enzymes like carboxylases, hydrolases, lipases, proteases, all these enzymes which are hydrolytic enzymes present inside the lysosomes. And these enzymes are active at acidic pH, not at basic pH. So, statement 1 is true and 2 is false. Fine. Next. See, this is also from Cell the Unit of Life chapter, wherein statements are given, and as I told you, whenever statement based question is given, you have to look into whether he is asking you to identify correct statement or to identify an incorrect statement. Right. So, now you see, quasi-fluid nature of lipids enable lateral movements of proteins, true. Okay. In fluid mosaic model, which is widely accepted, which is proposed by Singer and Nicholson in the year 1972, wherein in quasi-fluid nature of lipids, wherein lipids are present like this, which are floated, fine. Quasi-fluid nature of lipids, proteins are present as icebergs on the sea. This protein's lateral movement is possible because of quasi-fluid nature of the lipids. Mitochondria and chloroplast constitute endomembrane system, completely wrong, because endomembrane system consists 4 organelles which are single membrane bound. Recall what are those 4 organelles? Endoplasmic reticulum, endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. All these are 4 organelles which are single membrane bound, they are placed in the endomembrane system. Second is phosphate granules are not membrane bound, wrong. This is correct, because these are inclusion bodies. Inclusion bodies are the reserve food materials which are present in a prokaryotic cell. So, phosphate granules are non-membrane bound organelles which are present in a prokaryotic cell. Fine. Next, centriole is a component of all cells, wrong. Centriole is present only in the animal cells. It is present only in the animal cells. Centrioles are absent in plant cells. Fine, clear beta? This is all about this question. So, he asked us to find the correct statements. Correct statements are option 1 and option 3. Fine. Next, if we have to look into, this is again a match the following type of question from biomolecules. Glycosidic bond is formed usually in between 2 monosaccharides, and if you take DNA nucleotide, it is glycosidic bond is formed between sugar and nitrogen base. Okay. So, glycosidic bond is formed by dehydration of water molecule. Okay. The bond is formed between phosphate and this is ester bond. This bond is formed by dehydration. Okay. So, this bond is formed by what ma? Dehydration. Fine. So, glycosidic bond. Next, hollow woven ball folding is what? Tertiary. 3D structure of protein is found like how? Hollow woolen ball like structure. Next, enzyme. Here what enzyme is given in list 2? Trypsin is one of the enzyme. Next, ester bond. Ester bond, if you see, if this is a sugar molecule, carbon 1, 2, 3, 4 and 5. Okay. So, At carbon 1 what is attached? Nitrogen base is attached which forms a nucleoside. This all of us know beta. At 5th carbon, fine, what is attached? Phosphate group is attached. At 5th carbon, phosphate group is attached. This is by what? Phosphoester bond. This is by phosphoester bond. Okay, clear? So now yes, this is by what? Bond formed between phosphate and 5th carbon, that is phosphoester bond. Next. So this is how we have chosen it. Next question beta. Yeah, arrange the chromosomal events in meiosis. See, all the times whenever I am discussing, I told you definitely you will get a question from meiosis. That too mostly you are getting a question from prophase 1 of meiosis. Here is again a question from prophase 1 of meiosis. Okay, this is again from prophase 1 of meiosis. Wherein from prophase 1 if you see, okay, what are the 5 stages you have? Leptotene, zygotene, pachytene, diplotene and diakinesis, right? So first you see beta, in you see the events that are happening. Disappearance of nucleolus takes place in the last substage. Fine. So you have leptotene, zygotene, pachytene, diplotene and diakinesis. So if you clearly look into beta, in leptotene what happens? Pyrene stage is observed where chromatid threads will be attached to the nuclear membrane. Next, in the zygotene stage what happens? Pairing of the chromatids takes place, formation of synaptonemal complex, tetrad, bivalent chromosomes are observed. So here you see pairing of chromosomes happens in zygotene stage and appearance of recombination nodules takes place where? In pachytene stage where crossing over happens. So this occurs in the pachytene stage. Next, after this what happens? Dissolution of synaptonemal complex occurs in diplotene. Okay, in diplotene dissolution of synaptonemal complex happens. After the diplotene stage is terminalization of chiasmata takes place in diakinesis and dissolution of nucleolus also happens in diakinesis. So here these are out of amongst 5 phases 4 are given, fine. So now if you have to look into the options, first will be zygotene, pairing of chromosomes, second will be pachytene, recombination of nucleus, and third will be dissolution, that is diplotene. Next is terminalization of chiasmata and last disappearance of nucleolus at the end of diakinesis. Fine, terminalization of chiasmata at the beginning of diakinesis and at the end of diakinesis disappearance of nucleolus takes place. So always have an additional interest when you are looking into the meiosis. Definitely you will get a question from meiosis all the time. Now this is from anatomy of flowering plants, again important, look into it clearly. This is based on just your permanent tissues, ma. Okay. First if you see, mechanical support to the young stem and leaf petiole is given by what? Collenchymatous tissue. Second is what? Cells in fruit walls of nuts. In fruit walls of nuts what is formed? Sclerites. Third, cells absent in the primary phloem. In primary phloem what is absent? Bast fibers are absent. Endark primary xylem is stems vascular bundles. In stem vascular bundle, in stem endark pattern of primary xylem is seen. In root exarch pattern of primary xylem is observed. So now based on this you relocate the options. Next question if we have to see, yeah, again assertion reason question based on anatomy of flowering plants only. So here in assertion if you see beta, trichomes help in preventing the water loss due to transpiration. Yes, that is a correct statement. Reason he is saying the specialized cells near guard cells together constitute stomatal apparatus. We all know stomatal apparatus includes what? Stomata, guard cells and subsidiary cells. Okay. stomatal apparatus includes guard cells, subsidiary cells and stomatal pore. Fine. So, this is what is kept over here. Assertion, trichomes help in preventing water loss due to transpiration, that is correct, but reason is irrelevant to assertion. So, here what we have to choose, both assertion and reason are true, but reason is not explaining the assertion. Fine. Next. Here you see, phellogen made up of narrow thin walled rectangular cells, true. Phellogen is what, Nana? Cork cambium. This is made up of narrow thin walled rectangular cells. This is of what activity? Meristematic activity. Meristematic activity. Second, soft bark is formed in the early season, true again. More xylem vessels with narrow lumen are seen in the late wood, wrong. This is seen in the early wood. More xylem vessels are seen where? In the early wood. Okay. Next, isobilateral leaf mesophyll is differentiated, wrong. Isobilateral leaf is what? Seen in monocot leaf where the mesophyll is undifferentiated. Dorsiventral leaf which is seen in a dicot leaf, their mesophyll is differentiated into palisade parenchyma and spongy parenchyma, right. So, now we have to go with the options 1 and 2 because he asked us to choose the correct statements, right. Next, this question if you have to see, ma, this is from ecology, okay, wherein clearly the description is given, we have to go with the right choosing of options, that is it. So, here if you see annual plants which are found in dry weather, annual plants which are found in dry weather, this is the example from xerophytes, xerophytic annuals, that is tribulus. Second, submerged in water, submerged water plants with rooted, example is utricularia, no contact with water on the surface. Surface whichever are present, you know, salvia, free floating hydrophyte. Next example, perennial plants which withstand the drought conditions under xerophytes, best example is what? Casuarina. Okay. So, here we have to go with these options, annual plants with dry weather tribulus, submerged in water, submerged plant, that is utricularia. Next, no contact, that is free floating plant, salvinia. Next, perennials which withstand for many years in drought conditions, that is casuarina. Next. See, here this is a direct statement again from the ecosystem only. 1 acre of trees annually consume the amount of carbon dioxide equivalent to that produced by driving a car 26,000 miles, for 26,000 miles. The same acre of trees also produce enough oxygen for how many people to breathe for a year? Direct statement, that is 18 people for 1 year. There is no calculation involved in this. Now, assertion reason type of question again from transport in plants, plant physiology unit. Look into it carefully. Transport of, sorry, this is from plant growth and development, plant growth regulators question. Look into it carefully. Transport of plant growth regulators are always strictly polarized and unidirectional, he is saying. No, transport of plant growth regulators only auxin is unidirectional. It is polarized and unidirectional from shoot to root. Okay. Whereas gibberellins, abscisic acid, these are bidirectional because they also transport through phloem from source to sink. Okay. So, assertion will be wrong here. Next, organic and mineral nutrients undergo multi-directional transport, reason is true. So, here we have to go with the option assertion is false and reason is true. Next question beta. So, here choose the correct statements amongst the following he is saying. We have to go with correct statements. This is about again from transport in plants only, and this is from apoplastic and symplastic movement. So, here with respect to transport we have to go with what he is asking us, apoplastic and symplastic movement. Apoplast and symplastic movement. We all know apoplastic movement is from cell walls and it is intercellular. So that is how the movement of water takes place, and this is the pressure gradient is driving the water pump or water flow in the apoplastic movement, whereas symplastic movement is through plasmodesmata. Okay, this is through plasmodesmata and this is intracellular. Now you look into the statements properly. Movement of water through apoplast involves crossing the cell membrane. Wrong, because it is from cell walls itself. Movement of water through apoplast is dependent on the gradient. Is it dependent on gradient? Yes. Okay. Next, in symplast, water travels in cell through plasmodesmata. That is true. Transpiration pull has lesser significance than root pressure for water transport. Wrong. Transpiration pull has more significance when compared to root pressure, not less significant. So this is wrong. We have to go with options 2 and 3, right? Next. This is from nitrogen fixation from the chapter mineral nutrition. Look into it carefully. To answer this question, you have to be aware of the complete formula, last formula which is given for nitrogen fixation. So from one nitrogen molecule, how many protons are utilized? 8 protons plus 8 electrons, and 16 ATP are utilized to fix 2 ammonia molecules. One nitrogen to fix into 2 ammonia molecules, this many are utilized, plus 16 ADP and inorganic phosphate, right? Now he is asking a question about number of ATP, protons, electrons required for formation of 6 ammonia. So the question is for 6 ammonia. So here how we have to calculate? First let us do for number of ATP. 16 ATP are to fix 2 ammonia. To fix 6 ammonia, how many ATP are required? Okay, it is 6 into 16 by 2. Okay, 6 into 16 by 2. It is nothing but it gives you the answer of 48. Okay, it is as simple as it is. It gives you the answer of 48. Next, protons. Protons and electrons, how many are utilized to fix 2 ammonia? 8 protons and electrons are used to fix 2 ammonia. How many protons and electrons are utilized to fix 6? 6 into 8 by 2. Okay, it is 24. So what we have to go with? 48, 24, 24. That should be our option. 48, 24 and 24, right? Next question. Carefully answer. This is directly question asked from mineral nutrition only. Loss of chlorophyll leading to yellowing of leaves. Yellowing of leaves is what? Chlorosis, right? So there is option, direct question. Next question. This is from enzymes, ma'am. Enzymatic reactions. Exothermic reactions, P is at lower level than substrate. Product is at lower level than substrate. True, because in exothermic reactions what happens? Energy is released out, right? So in exothermic reactions what happens? Energy is released out. So product is at lower level than the substrate. Second, enzyme activity can be affected by temperature. True, with alteration of tertiary structure of proteins. Correct. Next, low temperature denatures the protein. Wrong. High temperature will denature the protein. Ligases can remove the groups from substrate. Wrong. Ligases will bind the groups from the substrate. Lyases will break the groups from the substrate. So now we have to go with the correct statement. So that is 1 and 2. Next question, beta. Yeah. Phenomenon that describes the first action spectra of photosynthesis. Here comes the interesting experiment that is given by E.W. Engelmann. Look into it properly. So this experiment says like, first, unilateral light when it is subjected to a filamentous algae, that is Cladophora. This filamentous algae conducts what type of photosynthesis? Oxygenic photosynthesis. So this conducts oxygenic photosynthesis, liberating out oxygen. To know the amount of photosynthesis observed, some aerobic bacteria were placed above this algae. This is the setup. Once unilateral light, first it is subjected to a prism. This prism will scatter this light to VIBGYOR. Okay. This prism will scatter the light to VIBGYOR, which is exposed on the filamentous green algae, that is Cladophora. Over this the aerobic bacteria are placed. Okay. So this aerobic bacteria, when it is placed on the green algae, at blue and red light region, you can observe two peaks after some time. Okay, two peaks indicating the accumulation of aerobic bacteria at blue and red light regions. So in these two regions, bacteria get accumulated, which means that maximum oxygen is evolved. So with this, E.W. Engelmann has drawn out action spectra and absorption spectra. Action spectra, which gives you maximum amount of photosynthesis conducted. Absorption spectra will give you the information about absorption spectra, that at different wavelengths, different pigments absorbing what wavelength of light is given in the absorption spectra. So these two are derived by E.W. Engelmann by this experiment. Now look into the question properly. Phenomenon that describes first action spectra of photosynthesis is elimination of Cladophora. Yes. Detection of oxygen release using anaerobic bacteria. Wrong. It is by using aerobic bacteria, right? Accumulation of aerobic bacteria in red and blue light region. True. Next, Cladophora in suspension of anaerobic bacteria. Wrong. It is in suspension of what? Aerobic bacteria. What are the two correct statements? 1 and 3. Okay, 1 and 3, that is option 4. So now you see, arrange the following intermediate substances of RUBP regeneration in correct sequence. So this is from third step of Kelvin cycle, that is regeneration, where it has 7 subsequent steps. So here if we have to go through, the very first product formed is fructose 1,6 bisphosphate. After that it is fructose 6 phosphate. Then formed is erythrose 4 phosphate. Then formed is what? Erythrose 4 phosphate. After that, biggest substrate which is there in the regeneration, that is pseudoheptulose 7 phosphate. After pseudoheptulose 7 phosphate, it is ribulose 5 phosphate and ribose 5 phosphate. Lastly, it is ribulose 5 phosphate. So what are in the order? First product is what, ma? Fructose 1,6 bisphosphate. Remember the products properly. Yeah. The first is fructose 1,6 bisphosphate. Second is, here I will write, it will be more clear. Second is fructose 6 phosphate. Third will be erythrose 4 phosphate. After that, pseudoheptulose, this is the largest organic substrate here. After pseudoheptulose, it is ribulose 5 phosphate, and last is ribose 5 phosphate. This will be the order, do remember. Next question is incorrect statement among the following. This is from what? Electron transport system, that is from respiration in plants. So from respiration in plants, if you have to see beta, electrons from NADH produced in mitochondrial matrix are by complex 2. Wrong. NADH is produced from what? Complex 1. Complex 2 is FADH2 or succinate dehydrogenase complex. Next, ubiquinone is oxidized to ubiquinol. True. Okay. After receiving from complex 1, that is NADH dehydrogenase. NADH dehydrogenase. NADH dehydrogenase. This will give out what? From mitochondrial matrix to the space this is from mitochondrial matrix to intermembrane space. Electrons are being pumped out, and here ferrous sulfur complex, FMN complex will be there. which is accepting 2 electrons, and this will give 2 electrons to ubiquinone, okay, which is a mobile electron carrier. This ubiquinone accepting the electrons, this converts to ubiquinol, fine, and then it submits the electrons to next complex, that is complex 3, okay, preceding to complex 1, complex 3 will be there, right. So, ubiquinones oxidized to ubiquinol, correct, ubiquinol oxidized to ubiquinone, this is wrong. So, amongst these 3, he is asking us to identify incorrect statements only. Incorrect statements are 1 and 3. 2 is correct statement because ubiquinone, which is a mobile electron carrier, is converted to ubiquinol. Next question, ma. Choose the correct statement among following he is asking. This is from again plant hormones only. So, abscisic acid, this plays important role in dormancy, not in stratification and scarification. Okay, this plays important role in what? Dormancy. Gibberellic acid will play major role in scarification, stratification. For seed germination, decapitation results in the growth of lateral buds. True, decapitation of apical buds will help in the growth of lateral buds by removing what phenomena? Apical dominance. Next, kinetin naturally occurs in plants. Wrong. Zeatin is what naturally occurs in plants. Zeatin is natural cytokinin, not kinetin. Kinetin is synthetic form of cytokinin. Next, vernalization promotes flowering at high temperature. Wrong. Vernalization promotes flowering at low temperature. So, here in the question what did he ask us? Correct statement to identify, that is option 2 only. Next. This is directly from the examples itself. So, Begiotova, and this is from biological classification only, that is from Monera. Begiotova is a chematotroph, Salmonella is a parasite, Rhodopseudomonas is saprophyte, Bacillus is Rhodopseudomonas is a photoheterotroph, Bacillus is a saprophyte. Next, this is from virus, from our Telugu Academy. This is also a direct statement given by virus. ICTV system, AIDS virus belongs to the genus lentivirus. Looking at AIDS virus, I think most of the students opted for HIV, but no, that is belongs to what? Belongs to the genus lentivirus. Next, here is the question from genetics, wherein in which of the following crosses both phenotype and genotypic ratios at F2 generation are 1:2:1 ratio. So, 1:2:1, red flower crossed with white flower in snapdragon. This shows what phenomena? Incomplete dominance. This shows incomplete dominance. Both phenotypic ratio and genotypic ratio is 1:2:1. Next, tall plants crossed with dwarf, this is monohybrid cross, there is no 1:2:1 ratio. Next, plant with dotted seed coat is crossed with spotted seed coat. This shows co-dominance beta. This shows what? Co-dominance. In co-dominance and incomplete dominance, what is the phenotypic ratio and genotypic ratio? Phenotypic and genotypic are 1:2:1. Fine. Next. So, here we have to go with the option 1 and 3. Homozygous plant crossed with heterozygous, that will not give you 1:2:1 ratio. Okay. Next question. Yeah. This is about purely scientists and their discoveries from molecular basis of inheritance. Look into it carefully. Hugo de Vries, he gave us about mutation. Alfred Sturtevant, he gave us about gene mapping. Morgan, he told about linkage and crossover concept. Sutton and Bovary gave us chromosomal theory of inheritance. Next question. This is directly from the table of molecular basis of inheritance again, wherein lambda 174 bacteriophage is having 5386 nucleotides in DNA. That is true because it has single stranded DNA. Bacteriophage lambda, it has this many base pairs, 48,502. Equally 4.6 into 10 to the power of 6 base pairs, true. Human haploid cell is having 3.3 into 10 to the power of 9 base pairs, not, sorry, 3.3 into 10 to the power of 9 base pairs, but diploid cell will have 6.6 into 10 to the power of 9 base pairs. Haploid cell will have 3.3 into 10 to the power of 9. So, as the wrong statement, we have to identify wrongly matched statement, so that is 4. Next, here again he is asking us to identify the incorrect statement. This is also from genetics molecular basis only, from replication point, okay, from replication concept. To answer this, once I have to give you this diagram of replication fork, okay, just a second. Yeah, in this if you see from 5 prime to 3 prime, and then leading strand is from 5 prime to 3 prime, and here this is 3 to 5 and 5 prime to 3, 5 prime to 3. This is lagging strand. So, now if you see beta, always do remember first replication fork is formed, and be it DNA synthesis or RNA synthesis, mRNA strand synthesis. Synthesis always takes place from 5 prime to 3 prime direction. So, be it DNA polymerases or RNA polymerases will act from which direction to which direction? 5 prime to 3 prime direction, right. So, now you see the small opening of DNA helix while replication occurs is what? The small opening is what? Replication fork. Second, DNA dependent DNA polymerase catalyze polymerization from 3 prime to 5 prime. Wrong. Polymerization or synthesis is one and the same, that is being from 5 prime to 3 prime. Here also you see lagging strand is from 5 prime to 3 prime. Next, the template of DNA strand with polarity 3 prime to 5 prime is leading. Wrong. The template strand is from what polarity? 5 prime to 3 prime only. This is not from 3 to 5. Next, DNA dependent DNA polymerase will catalyze the polymerization from 5 prime to 3 prime. True. So, here what are the correct statements? Sorry, 1, 4, 3 all are correct only, but the incorrect statement he asked us, incorrect statement is DNA dependent DNA polymerase will catalyze from 5 prime to 3 prime direction. And here the template of DNA strand with polarity 3 to 5 is leading strand. Yes, sorry. With respect to leading strand 5 prime to 3 prime, the polarity of template is 3 to 5 only. Okay. With respect to leading strand, you observe the polarity is 3 to 5 only. Okay. This is true, correct statement only, and the only incorrect statement is option 2. Next. Yeah, this is about, this is from biotechnology. Look into it clearly. A group of enzymes that removes the nucleotides from the ends of the DNA, that is exonucleases. And another group of enzymes that make cuts at specific locations within the DNA molecule, at specific locations. Those are what? Endonucleases. So, at the ends it is exonucleases, at specific locations it is endonucleases. So, it is 3 and 4. Next, methods which help in insertion of recombinant DNA in the host. How recombinant DNA can be injected into the host? It is not by selectable markers, it is by disarmed pathogen vector. Yes, by pathogen vector it can be done, by biolistic method, yes, and by micro injection. By these 3 methods, the recombinant DNA can be inserted into the host. Okay. So, these 2, 3, 4 are accepted. Next, choose incorrect statement among the following sentences. This is also from biotechnology only. Human insulin is made in fungus. Wrong. Human insulin is made in Escherichia coli, not in fungus. Okay, this is not in fungus, this is from E. coli. Next, insulin DNA sequences are introduced into genophore of E. coli. E. coli is a bacteria. From a bacterial cell we all know genetic genophore genetic material, main genetic material is present, and plasmid DNA is also present. Okay, fine. So, the genetic material is inserted where? Into the plasmid DNA. It is inserted in plasmid DNA, not in the genomic DNA. That is inserted where? Into the plasmid DNA. Next, insulin DNA sequences are introduced into the plasmid DNA, not to the genophore DNA. Recombinant E. coli produce insulin chains. True. Okay, recombinant E. coli will produce insulin chains. So now we have to identify the incorrect statements, that is 1 and 2. 1 and 2 are incorrect. Next, last 3 questions left, ma. It is not afference towards biosafety of genetically engineered crops. Again from biotechnology only. Direct statement from your Telugu Academy, wherein according to this statement, the direct statement which is related to biosafety of genetically engineered crops is unauthorized use of bioresources by multinational companies. Fine. So this is about that. Last 2 questions. This is from strategies of enhancement of food production. Pusa Gaurav, which is related to what crop? Brassica. Okay. Pusa Gaurav variety is against what? Nematodes. Next, as such we have Pusa Sembine. Okay. That is against what? Jacids, aphids and fruit borer. Pusa Swami and Okra. This is against aphids. Okay. That is against aphids and yellow mosaic virus. Sorry, that is against yellow mosaic virus and stem and fruit borers. This is also directly from table only. Table of strategies of enhancement. This is from microbes in human welfare again, wherein in detergents lipases are used, bottled juices pectinase is present, clot busters enzyme used is streptokinase, immunosuppressive agent is what? Cyclosporin A. Okay, immunosuppressive agent cyclosporin A. Next, methanogens, which are under archaebacteria, again from biological classification, where they are anaerobic and they produce methane from cellulosic waste. Okay, that is present in the gut of ruminants like cow and buffalo, where they will be producing what? Methane gas. So with this we have finished the complete botany paper, and stay tuned for more such questionnaire sessions which we are taking over here. Thank you.